certificate, the student will get a $25 gift card for being the overall people's choice. So without me wasting much more time, here's Erica, our first speaker. And I guess while she's walking to the stage, I'll be in control of the slides, and that way we don't have to worry about technology in our 3 minutes starting. So once she starts, I'll make a timer, and then you guys will see a slide that looks something like this, or exactly like this, and that'll be for the speaker to stop talking, and if they keep talking into this slide, they'll be disqualified from winning. So there's the rule. It's for the 3MT. I don't know what that is. Hopefully that doesn't happen all the time. But yeah, so here's Erica, and once she starts, I will start the time. Here's a pointer.
The average American family throws out around $1,300 to $2,300 worth of food annually. With this amount of food waste comes the waste of our nation's resources. American agriculture uses 10% of the energy budget, 50% of our lands, and 80% of our fresh water. So why are we wasting so much of our food? There are multiple reasons for this, but one of the big reasons is the food simply went bad. This food spoilage is associated with microbes that can cause post-harvest disease. So what can we do as plant scientists? Well, we decided to hypothesize to apply induced resistant elicitor before harvest that could prolong the shelf life of our produce after harvest. Now, induced resistance is comparable to the human immune system. We apply an elicitor to the plant, then this activates the plant's natural defenses and helps protect the plant against further infection. This has been commonly used as a pre-harvest treatment, but we want to evaluate its efficacy in use in post-harvest disease. Now, we use a variety of these elicitors in our treatments, including azelaic acid, salicylic acid, hypochloric acid, and glycerol. For comparison, we also included these conventional and organic standard fungicide treatments. And additionally, we included Actigar, which is a product currently on the market that works through induced resistance. Now, we apply these pre-harvest treatments throughout the growing season and then harvested and stored our tomatoes according to grower standards. We then inoculated our tomatoes with a calototricum that can cause the fungal disease ripe rot, otherwise known as anthracnose, of tomato. Now, this graph here shows the percentage of diseased fruit for each of our treatments. As you can see, we only had one treatment that shows statistically significant difference from our control, and that is our organic standard. We suspect that this is due to one of the products that we included in our organic treatment. Now, although we did not see efficacy with our elicitors in our post-harvest study, we did see differences in our mycelial inhibition, and we want to continue to look at these elicitors as a possible pre-harvest treatment to prevent post-harvest disease with different concentrations and application timings within our field, and hopefully reduce food waste. Thank
you. All right, so we have a built-in time schedule to allow our judges to finish their score in between. There's 2 minutes in between, so I would imagine it's very hard for a judge to listen to a presentation for 2.5 to 3 minutes and then score it based on only that. So we figured that since we're trying to start an event that would encourage, I guess, collaboration and other things, that would use this time for socialness, or social time in between. So if you guys want to talk, mingle for 2 minutes, hopefully in your seat, that would be awesome. And we'll start our next speaker shortly. And also, if everybody will please keep their cell phones silent. And I will make one more announcement for the presenters. I failed to mention that Erica was perfect on her timing, but I will stand up with 30 seconds left, and then you will see your slide change at 0 time. So that's the 30-second warning. All right, so the first two speakers are a little different because they do need to catch a plot. So we're going to go ahead and introduce our next speaker, Madison, who will be talking about birthing. Sorry if that wasn't enough time on the first one, Judge.
An apple a day may keep our doctor away, but what keeps the apple's doctor away? Apples suffer from many different diseases, but one of increasing concern in southern apple-producing regions is the disease bitter rot, caused by fungi in the Ascomycete genus Colotoxicum. Bitter rot causes the fruit to rot in brown, conical-shaped lesions, making the fruit unmarketable and decreasing yield. In Kentucky, during the 2015 and 2016 growing seasons, bitter rot caused an average yield loss of around 30%, with some reported losses as high as 90%. Colotoxicum species also cause anthracnose fruit rots of strawberry and blueberry. And with mixed fruit orchards being common in Kentucky, containing plots of apple, strawberry, and blueberry all within the same orchard, this begs the question, are the same Colotoxicum species causing disease on all three fruits? In an effort to answer this question, I am investigating the possibility of cross-infection within mixed fruit orchards, as well as seeing where the fungus is capable of overwintering. Multiple Colotoxicum species can cause bitter rot, and the specific species distribution present on apple in Kentucky has previously been studied. So I'm focused on understanding the species distribution on small fruits to see if there's a correlation between the two. So far, based on fungal culture morphology and PCR-based species complex identification, the possibility of cross-infection cannot be ruled out. Furthermore, lab cross-inoculation studies on apple, blueberry, and strawberry show that isolates are capable of causing disease on fruits from which they were not originally isolated. This also supports the hypothesis of cross-infection. The next step is to look at clonal variation within the most prevalent Colotoxicum species causing bitter rot, C. fiorini, using both telomere fingerprinting and PCR-based approach to see if the same clone is able to move between apple, strawberry, and blueberry within an orchard. The other component of my project focuses on overwintering. Not much is concretely known about where the fungus overwinters. So I'm surveying potential overwintering sites and identifying alternate hosts by taking tissue samples year-round from various apple tree parts and surrounding leaves to see if any Colotoxicum isolates can be recovered. In order to keep the apple doctor away, we need to know more about the epidemiology of bitter rot, so that ultimately growers can utilize more informed and effective management strategies. Thank you.
All right, so now, Bob, going on your time, our next sort of you guys have safe blocks wherever you all go. Can everybody hear without the mic? Because we have a portable mic that we can give the speakers that's up there as well. Or if future speakers want to stand behind the podium, we can have that mic on. That might make it a little easier for everybody. Yeah.
Yeah.
I mean, I think
All right, so to keep on our schedule, we have our next presenter, Matt Hobb.
I'm a big fan of collaboration. I think in a rapidly changing world, we can better accommodate for problems by increasing our perspectives and bringing in new ideas. My research looks at a natural type of collaboration between a grass, tall fescue, and a fungus that lives within it called Epicloe xenophylla, which I'll call Epicloe for short today. One way you can think about this interaction is to think about energy drinks and humans. Imagine an individual tall fescue as an individual human being. And Epicloe has several different strains, and you can think about them as different flu strains. It's the same infection, but different variations. And I've depicted this as 3 different flavors of an energy drink. Again, same drink, but different flavors. And if you drink a certain flavor, you get an associated resistance trait. So if you drink the first one, this will increase your biomass. You can outcompete smaller, weaker competitors. If you drink the second one, this will give you disease resistance. You can bolster your immunity. And the third one will give you drought tolerance, so you don't need as much water to survive. Now, let's say we have a population of just ones. This can be advantageous up until a certain point. Let's say there's a period of famine and a disease outbreak occurs. This is potentially very damaging for a population with no disease-resistant individuals. Alternatively, let's say we have a population of ones and twos. The same disease outbreak occurs. However, this time, our population has a better chance of survival. Why? Because now half of our population has disease resistance, we are increasing the range of conditions that our population can accommodate to. What if we continue increasing the different resistance traits present within a population? What if we had a population of ones, twos, and threes? And this gets to the core of my research. We predict that if we increase the different strains of Epicloe present in a population of tall fescue, we can increase long-term ecosystem sustainability. And tall fescue is not only environmentally, but economically dominant crop. It covers over 15 million hectares in the US. So it wields quite a bit of significance on a lot of large industries like cattle production. But also, tall fescue is just one of thousands of agricultural crops. We can take this kind of research, this kind of ecological assessment, and apply it to different food systems as we work towards energy efficiency. Ultimately, by diversifying our collaborations, we have the potential to increase long-term human survival. Thank you.
I do not have But we can start doing like a popcorn joke thing. Somebody gets called on, tells a joke, and we'll call somebody else. All right. What do you call a cow with 2 legs? Lean beef. What do you call a cow with no legs? Ground beef. So I have 3 kids.
The jokes do make it a little tougher.
The judges requested not to.
Either that or earplugs.
If I stand up here early,
Sure.
No, you get 2 more minutes.
This is going to be like one of those shows where we get to buzz you. So that was the original idea. We were just going to have a slide that pops up something obnoxious and then we just the stop speakers right there.
I'm glad you can do that.
Yeah. All right, well, to maintain the schedule, our next speaker is Dr. Haramoto.
So herbicide resistance has been called a game changer in the field of weed science. And resistance is an evolved trait in weeds, in which they have they are no longer able to be controlled by an herbicide that used to kill that weed. So this figure is illustrating a potentially, or an especially troublesome aspect of resistance, that is multiple resistance. So we're looking at the increase in populations that are not controlled by more than one type of herbicide over time. And we've got more than 50 populations worldwide now that are resistant to more than 2, or at least 2 types of herbicides. We've got about 15 populations that are resistant to 3 types of herbicides, and about 10 that are now resistant to 4 types of herbicides. This is classified by the herbicide site of action, and we only have this is how herbicides kill plants biochemically. We only have a limited number of these types of actions. But growers keep thinking that we're going to keep getting more and more types of herbicides and more sites of action developed, that they're just being or held up in development. That's not true. So we need to be developing cultural practices that we can use with herbicides to forestall the evolution of resistance and improve the longevity of the existing products that we do have. So one of those is the use of cover crops for weed management. So cover crops, either while they're growing, as you see here on this photo on the left, or after they're dead and after we plant our cash crops into their residue, as you see in this photo here on the right, can help with weed management, with and without herbicides in a number of ways. So they can outcompete weeds, they can keep weeds from emerging, and they can reduce seed production in weeds. And these are 3 ways that we know through modeling can decrease the evolution or forestall the evolution of resistance in weeds and reduce the spread of evolution. So my group is working with a number of different aspects of cover cropping to reduce the evolution of resistance. So we work a lot with the mechanics of cover cropping, seed selection, seeding rates, cover crop planting times, cover crop termination times. We work a lot with cover crops after they're dead already. So how cover crops influence crop establishment, crop growth, crop yield, like you see here on the photo on the right. We also look at how crop residues influence the use of soil residual herbicides. These are herbicides that are applied to the soil and keep weeds from emerging in the first place. We also look at how cover crop residues influence foliar applied herbicides. These are ones that are applied to weeds after they emerge and kill their foliage. So we have a number of different trials throughout the state and with collaborators in the region to try to, again, improve the longevity of the herbicide products that we already have and try to reduce the spread of resistant weeds. At this point, I'm just going to keep talking because I really want that to pop up. Princeton, we can hear you, FYI. You might want to mute yourself. I don't think you're allowed questions during the 3-minute meeting.
So, as a reminder, the judges were not judging the faculty members per se. However, everybody in this room is judging every speaker because we have a vote box in the back for your favorite faculty and your favorite student. At the end of the presentations, you can cast your vote. We'll tally them, and the faculty member will have a certificate or bragging rights. So, also. Yes, so Andrea Key is not able to make it. However, Early Amos has volunteered to take her place to help with scheduling, and he had a meeting come up anyway. So here in a minute, we'll let him take over. All right. Or actually, we're not judging the fact that we go ahead and start.
Okay, well, yeah, we can get ahead. Get ahead.
Lunch is on the way.
Hungry people.
So,
Yeah. You're it's a lot. It's pretty great. All right, so I'm in pursuit of cellulose biosynthesis inhibitors. And how we started this pursuit was through the ordering of a large compound screen, or a large diverse set molecule library that looks like absolutely nothing up here. And we screened it using a liquid handling robot. So we essentially just programmed what we wanted it to do, and we walked away. So we weren't bothered by the thumb, corrupting dilution monster, and we also kept our sanity. It was quite nice. And at the end of our dilutions, we started observing phenotypes. So this is an example of a bleached phenotype, and this is an example of a short root phenotype. We've observed multiple phenotypes. We categorize them in about 6 different categories. And while we were simultaneously doing these phenotypic categorizations, we were also growing a mutagenized population of Arabidopsis in the greenhouse. And the reason we grow this mutagenized population is so that we can find resistance for these compounds later on down the pipeline. In addition to observing our phenotypes, those phenotypes that we found that were very interesting, we did a chloroglucinol stain on. And what that does is it detects the presence or absence of lignin in your sample. Now, we screened 4-day-old seedlings, so 4-day-old seedlings should not have any sort of lignin present. However, some of the aberrant phenotypes, or the very short phenotypes, had it seemed like some meristematic or centrally located lignin. That to us is indication of a CBI. So we had about 33 compounds that produced this phenotype. So from there, we've been doing some confocal imaging to determine how these compounds influence cell shape, and additionally how these compounds influence the cellulose synthase complex. So right here is a normal plant cell, pretty well structured. These are all pip lines, so these are proteins that are embedded within the plant cell membrane. And these are also pip lines. And as you can see, there's a pretty aberrant cell shape. They're very non-anisotropic, so they grow in all directions. There's no directionality to it. And over here, this is me attempting to get some exposure to imaging sese through time-lapse images or creating movies on a confocal. And this is multiple images laid on top of each other, and you can see the cytosolic vesicles traversing underneath of the membrane-bound seses. So as we continue this research, fingers crossed, we'll find a novel compound that will inhibit cellulose. Thank you.
All right, so since we do have people down in Princeton listening, please stay through the post. That way they can do the presentation. And it's being recorded, I think, or something like that. So we can work that out. Thank you so much.
It seems more than 2 minutes.
That's true. All right, let's go ahead. Are you ready?
Have you ever heard about having high quality food, buying in the store with low price? To have a high quality food, we need to have a good field management. Good field management needs to be with integrating soil, crop, and agrochemical with regard to the rainfall in one process. However, it requires many field measurement, and sometimes it's not possible. That leads us to use agriculture system models. Agriculture system models are good tools for field measurements with a large input of soil, crop, and environmental conditions. With the agricultural system models, we can, with the agricultural system models, we can grow crops in a computer. That means We can use a computer to simulate all soil and above ground during the growing season. For example, the high and deficit nitrogen on normal long period of rainfall decrease and increase temperature, and we can have output for any day during the growing season with a high resolution. Of course, there is no free lunch. We need to provide a high quality input. We need to calibrate the model or adjust it to our field. With a good experimental design, solid calibration can be accomplished, and the successful calibration does not have to repeat it every year. That means the model can be used without any further calibration. So models best help to increase yield, water use efficiency, and fertilizer use efficiency, whilst the environmental harms can be minimized. In conclusion, the field measurements with agriculture, the field management with the agriculture models helps to obtain the high quality food that we are expecting to see. Thank you.
Does this mic work? No. How's this mic? Is anybody else going? Oh, there we go. So if you're a speaker, please use this if you plan to walk across the stage, just so Princeton can hear us. Thank you. We're just another minute to finish up. Dr. Dorbrand will be our next speaker. I do like the idea of having jokes, but just wait a lot of
So far, cannabis has been classified into 6 distinct chemotypes. Chemotype 1 has THC prevalent. Chemotype 2 has similar amounts. That's the second. Wish I had a chemotype 2 shows mixed levels, and chemotype 3 CBD is prevalent. Chemotype 1 and 2 generally have more than 0.3% THC, so they're considered marijuana. The other chemotypes are industrial hemp. They're not marijuana, and that's what we're growing here in Kentucky. And there's also chemotype 4, where the precursor of THC and CBD is prevalent, so it doesn't make these molecules. And then there's ones that don't make any of the precursors, and then there's ones that were predominantly make CBC. So hemp is a dioecious species. Males are XY, so these XY, and females are XX, just like in mammals. Very unusual in plants. And but different than mammals, the Y chromosome is bigger than the X chromosome. And one can induce female flowers on male plants, and this shows the induction of male flowers on female plants. So if you self a plant where you induce male flowers in a female plant, you get all female seeds. And so there'll be XX and vice versa. So this leads to, so just in, to finish, there's a lot of interest in industrial hemp in Kentucky for fiber, for seed, and for cannabinoids for medicinal uses. And it's found that THC oxidizes to CBN, and CBD oxidizes to a potent anti-cancer agent. But CBD has medicinal properties itself, particularly in treatment of epilepsy, but it can even be used to cure people or wean people off opioid addiction. Thank you.
This is the pointer, right? Okay. Okay. Hello, everyone. It is well known that plant viral diseases can destroy entire crops, causing huge economical losses and famine. Unfortunately, plant viral defenses are limited, and so it is our strategies to efficiently control viral diseases. In order to develop better antiviral strategies, we need to fortify our knowledge in plant viral replication. Plant viruses are intracellular parasites that exploit the host cell for replication by utilizing cellular resources, metabolites, and energy. In our lab, we work with tomato Boschiston virus, TBSV, and use a surrogated host model to identify the virus-host interactions important for replication. We have observed that TBSV, by stabilizing the active network, TBSV efficiently recruits host proteins, lipids, and metabolites to assemble the viral replication compartment. Moreover, these modifications in the host active network increases viral replication, as well as increases the energy demands in the cell. So, we have also found that TBSV is able to co-opt an ATP generating enzyme, PGK1, to assemble the viral replication compartment and provide the energy required at the replication site. Thus, my project investigates how the virus exploits the active network to efficiently provide the energy required to maintain the viral replication compartment. To understand that, I convert the levels of, using an ATP biosensor, I convert the levels of ATP in wild-type yeast and active mutant yeast with a stable actin filament. My results indicate that in the active mutants, the levels of ATP are higher compared to the wild-type. Furthermore, using an active mutant with depleted levels of ATP generating enzyme resulted in highly reducing viral replication. Together, this shows an intricate story where the active mutant and the ATP generating enzyme collaborate to fill the energy demands during viral replication. Understanding how and where the virus gets the energy for viral replication, we can create more specialized antiviral targets and help the pathogens to fight, the plants to fight viral pathogens. Thank you.
I still need some signals. So I don't want to be that guy either. Thank you for going on time.
Thank you, Mitchell, for giving me 10 minutes to my clock.
One of those things. Don't pay attention to it. All right, next up we have Dr. Amir.
Ignore that.
Oh,
Unintentional.
That's the animation.
Yeah, ignore it. I'm disqualified.
You're not really me.
Everybody hear me? Been a while since I've had a mic on the stage. When can I go?
Yep.
All right. Roots. Roots, folks, roots. Why do we neglect the roots? Show me some hands. How many of you do field research? How many of you work with plants in the lab? How many of you intentionally take data related to root system architecture, root function? Few. Why are we so neglected? Roots are important for anchoring the plant, right? For nutrient acquisition, for water uptake, and yes, for plant defense. Roots are important. More than 50% of that plant that you see in the field is below ground. The roots. The plant recognizes how important the roots are. They invest 20 to 40% of their fixed carbon below ground, releasing it as exudates to feed microorganisms to help them with defense, with nutrient acquisition. They're important. Why do we neglect them? Well, because what's hard to see is easy to neglect. It's like rust on the bottom of your car, right? It's there. It will cause a problem, maybe. But if you don't look, you'll never know. So that's where we're here to help. We're the Rhizosphere Science Lab. We're developing novel new technologies and building our own platforms like planar optodes to image the root's soil environment like pH and O2 and CO2 distribution in the root zone. We're building or have built a root phenotyping platform to look at root system architecture in 3 dimensions. So if you have a panel of wheat varieties that you're selecting for, you can go and see how that selection for above ground properties is influencing those below ground traits, so you can better predict how that new variety that you've selected will behave in the environment. We have zymography, where we can look at the distribution of enzymes in the 2-dimensional distribution of enzyme production, which tells us something about function. Mesocosms, we can take what we're doing in the field to put it into a mesocosm to look a little bit more like the field, but a little bit more controlled. And we have the capability of going to that field site that you're growing that corn crop on, or those wheat that you selected, and using many rhizotrons to go and image roots during the growing season out in the field. We are the Rhizosphere Science Lab. We pull all of these techniques together to address issues, concerns, problems, processes at the root-soil interface so that we can better improve the sustainability of agricultural production systems and understand better ecosystem processes in the terrestrial environment. Thank you.
So I guess it'll be up to the future PSA to determine if we want to change the rules of having animation. I stopped the other one with animation. Yeah, sorry.
I vote for animation.
Limited it. It's really hard on the field. A lot of grads. Our next speaker is Juliana. We're very appreciative that we had this interdepartmental meeting. It's very nice to team up with another department. So thank you all.
All right. So plants can defend themselves in a variety of ways. And one of them is through systemic acquired resistance, or SAR. So during SAR, signals are generated in this primary infected leaf. And these signals translocate using the plant vascular system to protect the whole plant against secondary infections.
So G3P is one of these signals, or glycerol triphosphate. So this signal is present in a lot of different plant cell compartments, such as mitochondria, chloroplast, apoplast, and so on. So the flux, the continuous flux of G3P in between cell compartments is very important to plant system community. Therefore, in my project, we want to identify proteins that modulate this flux of G3P in the plant cells. So in Arabidopsis, there are 5 isoforms of G3P putative transporters. They are known as G3P triphosphate permeases. So these proteins are also localized all over this cell compartment, and then they are also present in the plasma membrane cells. So we want to know if this the role of this protein, if this is actually able to translocate the metabolites during SAR. So I analyzed this plant G3P permeases for their transporter function, as well as for their contribution in plant system community. And indeed, these plant isoforms, they were able to transport G3P metabolite, or SAR metabolite during systemic cocoa resistance, and at least 3 of these isoforms were indicated to be associated with systemic cocoa resistance.
Is it working? Oh, yeah.
All right. Welcome, Muhammad.
Agricultural crops are affecting several environmental stresses due to unreliable weather condition. Temperature increasing due to global warming is one of them that affecting crop yield, especially seed growth and development. Seed development is very important not only to continue its heredity, but also it's important for good yield. Soybean is one of the most important crop of the world that producing for edible oil. It's also supplying food for human being, as well as it's using as animal feeding. Soybean seed development is also very important because it determines the total yield of the soybean. There are several stages of seed development in case of soybean. So the early or late phase is the very short stage, and during this stage, actually, it starts from the fertilization till the end of cell division, and seeds remain very tiny. However, after that, the seed filling stage is the time when seed become enlarged and determine its size. However, that's why several research work actually has been done in seed filling stage, but few research work also indicate that during the late phase, the temperature can manipulate the seed size. Therefore, in my proposed project, we would like to analyze the molecular and phenotypic analysis of seed development to understand what's happening if we put temperature during early stage of soybean seeds. Therefore, in my proposed project, we will grow soybean plants in different temperature conditions like normal or and low temperature and also increased temperature. After that, we will analyze those seeds and we will also check the embryo and endosperm development of early leg phase of soybean seeds. After that, we would like to use the laser capture micro dissection system that will allow us to separate different compartments of early seeds. And after that, we will collect the samples from the different compartments and we will use the sample for RNA-seq analysis. And from the RNA sequencing, we will generate the transcriptional profiling of early phase of seeds that will allow us to understand what's the molecular basis behind that. Therefore, we think our project will be very helpful not only for the molecular biologist, but also for the crop breeder to develop high yield soybean variety. Thank you, everyone.
Are we ready? A few more minutes? This is a finnick fox, an inhabitant of the Saharan desert. You can tell he lives in a hot desert by the extraordinary size of his ears. They help him dissipate heat during the hot Saharan days. His thick fur helps insulate him during cold Saharan nights. He is leaning against an aggressive, headstrong wind. Winds in deserts are dominant features and carry much energy capable of moving significant quantities of sediment, which you can see is happening near his feet here. His eyes are squinted in part to shield his retinas from the substantial UV radiation load that is irradiating both from above and the soil surface into those organs. What you cannot see is the sophisticated renal system he has developed to allow him to make use of every single drop of water he encounters. These ecological factors that have shaped the evolution of his species are also the factors that will dictate the decomposition of his body upon death. Globally, decomposition is controlled by temperature and moisture, such that decomposition rates usually increase with increasing actual evapotranspiration. Deserts have very low actual evapotranspiration because they have very little water to evaporate. Therefore, people have long hypothesized that deserts have very slow rates of decomposition. However, data from these systems show that they sometimes have surprisingly high rates of decomposition, leading ecosystem scientists to believe that some of these other ecological factors, rather than just water and moisture, are stimulating this significant process in these areas. My research program evaluates the effects of abiotic photodegradation resulting from radiant energy and soil litter mixing or soil deposition on the rates of decomposition in arid lands. Using a manipulative experiment in the Sonoran Desert, a 2x2 factorial where we have 2 levels of radiant energy, full UV and no UVB, 2 levels of soil coverage, no soil coverage and full soil coverage, I measured plant litter decomposition and microbial community over a year. What I found was that litter composition rates are highest under full UV and no soil coverage, and they're dominated by fungi. When soil coverage occurs, the microbial community switches dramatically to one of bacteria, and the overall process is slowed. Decomposition is the primary source of nutrients in these systems as they have very low organic matter. Deserts cover 1/3 of the terrestrial land surface and support more than 1/3 of human population. Understanding the factors that control release of nutrients and ultimately life is important. Thank you.
Thank all of our speakers in the first session. We're also having a lunch provided that should be out there right now, so please stick around, have lunch, and then we'll start the second session at 2 o'clock. Don't forget to come back and vote for your favorite speakers at the end of the second session.